This is Chapter 3, Module 5, Transcription, Translation, and Protein Synthesis. The learning objective of this module is to describe transcription, translation, and protein synthesis. Each cell needs proteins in order for it to survive. These proteins are used to replace old or damaged structural cell parts, to protect the cell, to carry things, to respond to changes, and to catalyze reactions. Basically everything the cell needs to survive on its own. New proteins must be produced or synthesized on a continuous basis. Every cell that contains a nucleus will be able to make its own proteins. Within the nucleus are the instructions on how to make new proteins. These instructions are invaluable to the cell. These are the only instructions the cell has for its survival, and so it's very important that they are kept protected within the confines of the nuclear membrane. These original instructions must never leave the nucleus where they could be possibly damaged or destroyed. The proteins, however, are produced outside of the nucleus in the cytoplasm of the cell. So, copies of these instructions will need to be made. These copies can move to the cytoplasm while the originals stay protected within the nucleus. These instructions are called DNA, and the copies that are made are called messenger RNA. The process of making messenger RNA is called transcription. In other words, the instructions were transcribed or copied so that the original instructions could remain in the nucleus, protected by the nuclear membrane, while the copy leaves the nucleus through the nuclear pores to enter the cytoplasm. In the cytoplasm, a new protein molecule can be produced. The messenger RNA binds to a ribosome, and the process of translation occurs where the messenger RNA directs amino acids within the cytoplasm to combine together to form a protein. In other words, the language of the messenger RNA will be translated into a sequence of amino acids. This module will discuss both processes necessary for the protein to be produced, transcription and translation. To understand these processes better, we should take a closer look at DNA. Each DNA molecule contains segments called genes. A single gene contains the instructions to make one protein. Within each gene is a specific sequence of nitrogen bases. Remember that adenine, thymine, cytosine, and guanine are the bases for DNA. Each gene contains thousands of these bases that are arranged in a different sequence in each gene. The sequence of these bases is called the genetic code, and it determines what protein the gene will produce. On each gene segment, the bases are grouped in threes called triplets. One triplet provides the code to produce one amino acid. Since proteins are chains of amino acids, each triplet will code for one amino acid in that protein. For example, if a gene segment has 1,200 nitrogen bases, that means there are 400 triplets and 400 amino acids would be produced. The resulting polypeptide, or protein, would be 400 amino acids long. Module 5 of Chapter 3, Transcription, Translation, and Protein Synthesis.